Morning, everybody. So Bone Therapeutics, as I said, we're a Belgium-based company. Our company was established in 2006. We are a cell therapy company, and we're focusing on bone diseases and some orthopedic conditions. Established in 2006, and we had the delightful event a couple of weeks ago. Actually, we went public February 6th on the Brussels and Paris Stock Exchange. So that makes us a public company as well. And the disclaimer, well, actually didn't get in the presentation, so it should have been there. So what is the ambition of Bone Therapeutics? We actually want to become a game changer in the field of orthopedics. And how would we want to do that? Well, the company wants to focus on bone repair and on fracture prevention. Why are we talking cell therapy here? Well, the skeleton is just like a number of other organs, a real natural regenerative process. It renews all the time again. And what we see in orthopedics is that, well, the current standard of care is always quite invasive, very, well, not very healthy for the patients to start with, not always very effective, and in some indications we don't see no solutions at all coming from the orthopedics. So our idea was, well, let's try to produce these cells that are actually responsible for rebuilding our skeleton in a factory and see if we can add that to this if we can add them in the regenerative process, in the healing process of where there is bone defects. What makes us unique, very briefly? Well, there's a couple of companies working in this space, but what makes us unique is that we're not working purely on stem cells. We decided to go a bit further, and we're working on osteoblasts. Osteoblasts are the bone-forming cells, which makes a lot of sense, as we believe these cells will be much more potent in the healing process, as all the injected cells will actually do the job of bone forming, and there's not like stem cells, which only will be partially doing that particular job. So more potent, and also we feel out of all the clinical trials, preclinical work we've been doing, that this is a much safer approach as well. Actually, osteoblasts arriving in different parts of the body, in different tissues, actually don't have any function anymore and simply disappear. So much safer. And then with this osteoblast, we actually went for another treatment paradigm within orthopedics. And you have a small example here where we're actually treating a patient with these osteoblastic cells. There was a patient here which has an osteosynthesis after a very severe fracture. And what we are doing is, instead of the standard approach, which is a bone autograft, which means you will remove a piece of bone out of the pelvis of the person, then actually replace that into the fracture zone, but you have to open the patient twice, very heavy surgery with lots of complications, long hospitalization. What we are doing is actually making a small incision at the height of the fracture zone here, bringing in the long needle into the fracture zone, injecting cells, removing the needle, closing it up, and see this is the bloodless. So that looks like really like a new paradigm within the orthopedics treatments. What are we looking at? Well, the markets we're looking at is fracture repair, and within fracture repair, we're talking about non-unions, delayed unions. Non-unions are fractures not healing after a period of 6-7 months. Nothing will happen anymore with these patients. We're talking about 300,000 patients, and delayed unions is a very interesting field, as there is about 1 million people after having had a long bone fracture with osteosynthesis or not having any effect 3 to 4 months after that particular treatment. And today these patients are not treated anymore. So quite an interesting field to work on. 1 million people that can be treated with a new solution with cells. And then in the field of fracture prevention, much larger markets, of course. Everybody knows osteoporosis. There we're looking at severe cases of osteoporosis, patients not responding to any of the current treatments. And that's 10 million patients over the United States and Europe. And then one other field, osteonecrosis, a very painful disorder of the hip, which always leads to a full replacement of the hip and comes with very young patients. An orphan disease, but still about 200,000 patients that we're looking at. Today we have, with our 2 product platforms, we have an autologous platform and an allogeneic platform. We're addressing 5 different indications which are in 5 clinical trials today. We have some preclinical work as well, but that's what the core focus is today. We are in these non-unions, as I said, into a phase 2b/3. We're also in osteonecrosis already in a phase 2b/3. And then we have started up 2 new trials, delayed unions, a trial in spine fusion and osteoporosis with the pre-op and the allogeneic project here, which are now in a phase 1/2a. And I think that's as an introduction. can bring us to Q&A.
Great, thank you, Wim. So I agree, it makes a lot of sense focusing on osteoblasts to build bone. You mentioned that your first program is autologous cells for non-union fractures. And I wanted to ask a little bit more about what your phase 2 experience was and what you learned from that study and what development plans are for pre-op. Is that the right way to pronounce it?
Pre-op, yeah.
Pre-op,
yes.
in non-union fractures.
So for non-unions, we had a very successful phase 2. What we've done, we treated about 28 patients who had such a non-union, and we were looking at an average period during which they had this non-union, non-healing after the trauma for about 2 years. Actually, there was one patient having this non-union for 11 years, and we're not talking about very old patients. In general, these are trauma patients who had a heavy car accident or a sports accident. So it's not because of age. It was a very widespread number of people, of ages of people we had in the trial. And out of this group of 28 people, actually 25 people healed, their fracture was healed. Only 3 didn't heal in the end for reasons which were, well, in fracture, if you have an osteosynthesis and you fall on it, it's quite, well, difficult that the fracture will heal after that event. It's like probably rebroken at that point in time. So 25 patients out of 28 were healed, and that made us decide to go into the phase 3. And the phase 3, there we have a very favorable design. We will have to treat 176 patients in that trial versus in a non-inferiority design versus the standard of care. And we hope that this will be, as successful as the phase 2 we had already.
And just to ask, what is the standard of care that you're comparing against there? And when do you think we could actually get data from that phase 3 study?
Yeah. Okay. Well, standard of care there is, as I came to explain, I tried to explain in the presentation, it's actually this bone graft, which means they will take a piece of bone out of the hip of that patient. They will crush that, and then that material that they have, which is, well, real bone of the patient itself, will be placed in the fracture zone. And then the patient, well, they hope that this will help the regenerative process for healing. In the 28 patients we treated, actually half of these patients had multiple of these procedures before, but this had no impact with them. So just adding the cells was much more effective, and that's why I'm saying it's a non-inferiority design versus the standard of care will be very helpful for us. And what was the next point?
Just when you think we might be...
Yeah, well, in this phase 3 trial will give us half of the patients will be treated during the course of next year and beginning of this year, and we hope to have interim results in the course of 2016 on this trial in Europe. And the idea is also to launch this trial in the United States somewhere in 2016.
And I'm going to go a little bit out of order from the slide that's up here and kind of move into prevention and stick with the pre-op process, product and talk about osteonecrosis and ask about how you identify these patients and what the plans are with this phase 3 study.
Okay, so osteonecrosis, just quickly repeating what it actually is. It is a painful disorder of the hip. Young men between the age of 30 and 50. If you had it, it's a fast progressing disease, and the causes for the disease need to be sought in, treatments with corticoids are very often referred to as a cause of this disease. Also, alcohol abuse is another one. But if you have a young man after the age of, at 30, you have a hip replacement, you need another one at 40, 45, and you might need another one at 60, and you can do it twice, but you can never replace a hip 3 times. So it's a very invalidating disease. So we are... There we had also a very successful phase 2 on this disease. We treated 63 hips, as it's a bilateral disease, but very often it comes on both sides for the patient. And there we actually have proven that we could reduce the fracture risk over a period of 2 years from, untreated being, almost 100%, that these people will fracture after a period of 2 years. If they treat it with the standard of care, which is just a cord decompression, which is drilling a hole in the femoral head of the hip, and those patients, well, there is a 66% risk they will have a hip fracture. In our trial, we've proven that it was 19% with the treatment with pre-op. So on that basis, we're moving on to the phase 3 now for treating 130 patients in Europe. The way we are identifying these patients, I refer to it's an orphan disease, and identifying patients in a fast-progressing disease sometimes seems to be quite difficult. So we worked on that together with rheumatologists to develop an algorithm in how to quickly identify these patients, but very often they go to the rheumatologist and they get treated with anti-inflammatory means, then they get an RX, and you can't see it on an RX. You need to do actually a scan to be able to see what the problem is with these patients. But based on the algorithm, based on the question list you have for the patient, you can immediately refer him for a scan, actually diagnose him in time, and work on the healing of this disease. And this has been seen very effective in the phase 2b trial in recruiting of patients and accelerating the recruitment of these patients. And next to that, we're working with patients as well. Patient groupings in Europe are not very common. We have them in the UK, so we're working closely together where we started our trials as well. And then in other countries, we're working to social media on that.
Very interesting. And it dovetails really nicely into the third indication for your autologous product, which is osteoporosis, which I find really, really compelling. How do you deliver the cells for this indication? And what are your ultimate partnering plans here? Because this is obviously getting to be a different type market, maybe a larger market than Bone Therapeutics could address on their own.
Absolutely. So osteoporosis, what we're doing there, as this disease is actually spread over the whole axial skeleton, just a local approach would be very difficult. So we decided to go with the pre-op product, being an autologous product for safety reasons, into an intravenous approach. We inject the cells, and what we are checking in this proof of concept trial we're currently running is if the cells are actually homing versus that axial skeleton. And further, we also look at efficacy. Will the general health condition of these patients improve after the treatment of drug? Will their balance be restored as we expected it should happen? And indeed, this trial is now on 20 patients. We can do that, but in the next phase, we'll go in for a partnership in Europe and potentially outside Europe.
Excellent. Now, what led you and Bone Therapeutics to migrate from autologous, from pre-op to an allogeneic cell-sourced product with ALLOP?
Well, it's a very logical step. We had to do a proof of concept with pre-op, and we've been doing that very successful. As we see the phase 3 trials, we are currently running on that. It brings, of course, advantages from a logistics perspective, also from a production perspective. Starting from a universal donor with the allogeneic project, we can do today out of one donation up to 100 batches of product. So that will reduce the production cost vis-a-vis the production of the pre-op product. It also will allow us to scale up production much easier and address larger populations, larger indications. And that was the main reason for doing it. We looked a lot of the safety issues before moving into that because, okay, allogeneic means healthy donor cells from a different person. We've done a lot of preclinical work, but the best proof of that the product is actually safe. Within the delayed union trial, we announced beginning of February that we had treated first cohort of patients, of 4 patients, safely and with the necessary efficacy on the delayed union with a proper healing of these patients. And we have more patients treated by now and actually no safety issues. Also not any immune suppressive agents which are used with the treatment.
So it makes sense. So it really enabled you to go into that larger delayed union market opportunity using the allogeneic cell sourcing. So we only have about a minute left, but I want to ask about this final program, which is in spine fusion. And I think you're using osteoblasts to help with disc repair. So what role do they play in that, and how can osteoblasts help with disc repair?
Yeah. Well, to make it clear, we're not working on disc repair. Actually, we are looking at spine fusion.
Oh, I'm sorry, yeah.
So that's slightly different. We're looking really where the discs are really severely damaged, and where the standard treatment would be a treatment where they would take away the disc and then place a cage with calcium phosphate granules, for instance, as a solution for this patient. And then that procedure normally should lead to a consolidation of two vertebrae. What we want to do here, as in Europe, the BMPs are not, well, have never been used a lot, and in the United States, well, there is some issues on that side, is that it's a very similar approach. The cells will be injected into that cage where the granules are placed, and that should accelerate the healing process to start with, as it takes between up to 12 to 18 months sometimes. And also in many cases reported, or over 25% of the cases are reported that the procedure is not effective. We hope with introducing the cells that the regenerative process to make the two vertebrae heal and to actually eliminate the pain for the patient will actually, result in the desired effect,
Great.
healing for the patient.
Excellent. Well, that's a very helpful overview. I know we could have spent some more time on this, but Wim, thank you for being here, and look forward to learning more and hearing more about these trials as they read out.
Thank you.